Hi guys, today we will explain identical reproduction and cell cycle. First of all, what we mean by cell cycle. Cell cycle is interphase plus mitosis. So for any cell to be able to make mitosis, it should pass by interphase. Now, which type of cells have cell cycle? The cells that have cell cycle are the cells that are able to make mitosis, and as we know, the cells that are able to do mitosis are the stem cells. Okay, so all stem cells have cell cycle, which are the somatic stem cells, the embryonic stem cells, and the germ stem cells. Now, for sake of what we make mitosis, أكيد for growth and development, for renewal and regeneration of cells. So it is an internal repair mechanism inside our body, and as well for the development of a zygote into a fetus. Now, this video I will send you the link for it. It will be helpful. It explains the cell cycle. I will explain it in the slides. Okay. Now, the first phase in the cell cycle, as you know, as we said, that the cell cycle is interphase plus mitosis. But in interphase, we have three phases. We have the G1 phase, which is gap 1 phase, the S phase, and the G2, which is the growth phase. Okay, we will start first by G1 phase, okay, which is the gap 1 phase. As you see, the duration of this phase is 8 to 12 hours. Okay, and as you can see that this phase is directly after mitosis. Okay, so at this phase the cell is leaving mitosis, the two daughter cells are leaving mitosis, and they will enter in G1 phase. Okay, so if you can remember mitosis, it is the division, it is the cellular division. Okay, so I'm resulted in a small cells, in two small cells, okay, that have the DNA, each chromosome of one chromatid. Okay, so these two cells, when they will enter the G1 phase, they still have two N chromosomes, and we discussed it before that two N chromosomes means that each I have two copies from each chromosome. I have two copies from each chromosome, and each chromosome is of one chromatid. So the amount of DNA here in G1 phase, it is Q. Now this phase, it is the longest phase. In this phase we have intense cell growth and activity, because as I told you, these two cells are small. They are resulted from cell division, so they have to go in intense cell growth and activity. The cells increase in size, where the cell volume doubles. All the organelles and the cytoplasmic components, including the centrioles, replicate. Okay, the cells prepare for DNA synthesis, so it synthesizes all enzymes required in the synthesis of DNA, which is the S phase. Now the DNA amount stays the same, as I told you, so it is stayed the same as resulted from telophase, and the amount of DNA is Q. Okay, the number of chromosomes is 2, and number of chromosomes, each chromosome is of one chromatid. So this is in G1 phase. So in G1 phase we have intense cell growth. The cell is preparing its organelles, it is preparing all the enzymes needed in the synthesis phase. Now, synthesis phase. In the synthesis phase we have DNA replication and synthesis. So I'm synthesizing the DNA. In G1 phase, I have each chromosome is of one chromatid. In the S phase, I want each chromosome to be of two chromatids. So I have DNA replication. There is duplication of chromosomes, where the chromatin of one chromatid is duplicated to become two. The DNA amount is increased to double, which is 2Q, as you see here. So the DNA quantity becomes 2Q. Two and number of chromosomes. We have two and number of chromosomes, but now they are of two chromatids. So each chromosome become of 2 chromatids. In gap 2 phase, which is the growth phase, we still have 2 number of chromosomes. So as you see, in all the interphase there is 2 number of chromosomes. No change in the DNA amount, that is it stays 2q. Okay, in this phase the cell is preparing for mitosis. Okay, it is preparing for cell division. So all the enzymes needed to aid in cell division are produced in G2 phase. Okay, now if we want to add up all the phases of interphase, we said that G1 phase, the duration of G1 phase is 8 to 12 hours. For S phase, it is 5 to 6 hours. And for G2 phase, it is 3 to 4 hours. So the total duration of interphase is about 16 to 22 hours. So look, it is 16 to 22 hours, whereas for mitosis, it is only 1 hour. Okay, so in general, cell cycle is the cell division. After cell division, the 2 daughter cells, they have to grow up, they have to produce all their organelles or their enzymes, or they have to duplicate their DNA in order to be able to start another mitotic division. Okay, this is the cell cycle. So as you see here, this slide summarizes all the info we discussed before. Okay, now and this is also, it summarizes all the information we discussed before. Now if I want to draw the graph for the DNA, for the variation of DNA quantity as a function of time in cell cycle, I have to take into consideration 2 factors that affect the amount of DNA. The 2 factors that affect the amount of DNA are the number of chromosomes and the number of chromatids, because I'm dealing in mitosis, I'm dividing the chromosomes into the sister chromatids in each chromosome. Okay, and in S phase, I'm duplicating the single chromatid to become a chromosome of 2 chromatids. Okay, so if I want to draw the graph, it will be like this. Okay, I want you to identify the phases of cell cycle on this graph and schematize a chromosome at each stage of the cell cycle. Now, the phases, as you see, this is all is interphase, and when we have a drop in the DNA quantity, okay, it is mitosis. Now, if I want to add the phases of interphase and the phases of mitosis on this graph, let me tell you that in this graph, to identify the phases of interphase, you have to consider the duplication of DNA quantity. As you see here, I have 7.4, okay, it is duplicating to become 14.8. Okay, the DNA quantity is duplicating to become 14.8. So in this, when I have this increase in the DNA quantity, it is obvious that this is the S phase, because I know that in S phase the DNA duplicates. Okay, so this will be the S phase. So this will be the S phase. And I know that before the S phase, I have the G1 phase. So of course, before the S phase, this is the G1 phase. And after the S phase, it is the G2 phase. Okay? So when there is an increase in the DNA quantity, it's obvious that this is S phase. Before S, G1, after S, G2. That's it. Now, mitosis, I will start mitosis here. Okay? Here, I still have, if you can remember mitosis, it's prophase, metaphase, anaphase, and telophase. In prophase and metaphase, I still have the chromosomes of two chromatids. Okay, the chromosomes are still of two chromatids. So prophase and metaphase should be here. Okay, prophase and metaphase should be here after G2, but they still have two chromatids. Each chromosome is still of two chromatids. But in anaphase, I have polar ascension. I have separation of the sister chromatids. So there should be a decrease in the DNA quantity. So this will be this decrease in the DNA quantity is the anaphase, and here we have the telophase. Okay? If I want to schematize a chromosome at each stage of the cell cycle, it's obvious that at G1 the chromosome is of one chromatid, because in these cells they are resulting from mitosis. Okay, so in this phase they are of one chromatid. In the S phase I have duplication of the of DNA, so the chromosome will be of two chromatids. In G2 it's still of two chromatids. Prophase, metaphase also it is of two chromatids. In anaphase we have polar ascension, so the two chromatids are separated, so the chromosome is of one chromatid, and at telophase it is also of one chromatid. Thank you.